Okay, just two sentences. Praveen is a PhD student in Tübingen at the Max Planck Institute. You're working with Klaus Scheffler, but this project is something where you're mostly being advised from Philipp Eses, who has then moved to Bonn. It is about the subject matter of many things that we care about, namely high-resolution imaging at ultra-high fields. Praveen is actually looking at 9.4 Tesla, I believe. Specifically with high resolution at ultra-high fields, we have these issues of the large training reins, which is why in Tübingen you are spearheading these other contrasts that are not great in EchoBolt, but balanced SSFP, which depending on the kind of parameters maybe you introduced this, are more microvessel sensitized as opposed to the large training reins. Please go ahead.
Yeah, thank you for the introduction. Yeah, so today it's going to be a little less fMRI and more acquisition, I believe. And so what we are going to talk more about is the SSFP bold, how it is different from gradient echo, and how we can combine this SSFP bold with spirals and ultra high field, what are the problems and stuff. So we talked briefly about the acquisition and reconstruction and some high resolution fMRI results. So yeah, most of the things are from the paper, but we'll start with some introduction about the SSFP bold. So as you know, we all want to detect neuronal activity, but we usually detect the bold signal, which is temporally and spatially blurred because of our biological neurovascular coupling, like hemodynamic response functions, and acquisition methods like a gradient echo, spin echo, different acquisition modes, and imaging parameters. So as a layer fMRI and high resolution fMRI people, we want the bold effect to be contained within our image voxel. So we want to make bold vessel specific so that you don't get contributions from macrovasculature in our high resolution fMRI. So we can either make bold vessel specific, explore other functional contrasts like vaso, or you can go into modeling this bold signal to reduce the macrovascular contributions. So like to start with, let's see what's spin echo bold, how it's different from gradient echo. So here you have a single voxel, which is a simulated voxel, which has a lot of different micro and macrovasculature. And the both vessels distorts this B0 field because of the susceptibility effect of the deoxygenated blood. So when you have such voxel, we are interested in the signal defacing, which is the differential signal we detect as a bold signal. So this signal defacing happens in two regimes, kind of. So one happens near the macrovasculature, which is called static defacing, and there is a dynamic defacing, which happens mainly near microvasculature. So the static defacing is a larger effect because it's a larger blood vessel and distorts the field more. And the dynamic defacing is a smaller effect, but the field is quite heterogeneous, like spatially very heterogeneous field. So so the white scribbly lines is the Brownian motion of the water protons, but in reality, it's much smaller than this. I can't zoom in anymore without getting blocky artifacts. So I'm take this as intuition rather than an absolute figure. So the gradient echo bold treats both defacing the same. So the stronger defacing gives a larger signal change, and the signal defacing from the smaller vessel gives smaller signal change. So this is the response of the gradient echo bold with respect to vessel size, but here it's a sphere, I guess, from this paper. And things gets interesting when you apply a refocusing pulse. The static defacing caused by the larger vessels are usually refocused because the spin stays in the same field environment. But in case of defacing, dynamic defacing, you have water protons which are diffusing all around the place with different fields. So you can't really refocus them. So spin echo bold in theory is like a diffusion contrast, but instead of applying a diffusion sensitizing gradient ourselves, the microvasculature will generate such a diffusion sensitizing field for us. So when you combine diffusion and a heterogeneous field, you get a spin echo bold, which is of course a smaller effect than a gradient echo, but have a much lesser macrovascular contribution. So now you know about spin echo, but we need to acquire it at a reasonable rate to perform fMRI. So the spin echo is very slow for fMRI, so we usually accept some form of T2 star contributions and go with one of these following methods. So different methods have different gradient echo, spin echo weighting. So we only focus on SSFP contrast. So the SSFP acquisition is quite simple. So you need a rapidly repeating RF pulses interleaved with some acquisitions, which can be gradient encoded. And at the beginning of every RF pulses, all the gradient moments has to be nulled. So here we are not applying any gradient, so you don't need anything. So this is the simplest sequence. You don't need any spoiler. It's just a rapidly repeating RF pulses. So we usually have a TR, which is the interpulse TR, which is different from a volume TR, which is for high resolution, it goes up to 4 milliseconds. So you repeat, acquire one line of K-space, and then repeat again until you're done with the entire K-space. So because we have to repeat this pulse very rapidly, most of the SSFP acquisition are 3D imaging rather than 2D. And so once you switch gradients on and off in your acquisition, you get some image like this, which looks like a T2 image, but it's actually a T2 over T1 ratio contrast. And it has one more peculiar feature, which is quite bad for us, but quite interesting as well. So the image intensity is sensitive to the frequency of resonance of your brain. So this gives you this low intensity regions at every periodic interval. This period is usually 1 by TR. So if you repeat your pulse much faster, then you have a more homogeneous region. So we try to keep our region of interest within this flat region, which we call passband. So we try to keep all our region of interest in this frequency band. So therefore we have a more stable activations. So as you know now, bSSFP, we don't use spoiling. So we have a lot of signals evolving simultaneously from the previous excitation. So the SSFP bold is not like a spin echo or gradient echo, which is restricted to one TR, but it evolves over multiple TR. This makes it quite complicated to think about. So if you have only think about SSFP, you can think about in different signal pathways. Some are like behaving like a gradient echo pathways, and some are behaving like a spin echo pathways. But in fMRI, you have a lot of things to worry about. So let's simplify this drastically and then have this, hopefully it's a simplified version. So when you have a RF pulse train, you have some steady state longitudinal magnetization and decaying transverse magnetization. Since you apply when you apply a low flip angle pulse, this different these two signal pathways generate even more signal pathways. So one is generated from the longitudinal magnetization. This is the excitation pathway, and this is like defacing pathway from the old excitation, which are undisturbed by the RF pulse. And you also have some refocusing effect. When you have a 180-degree pulse, all the transverse magnetization is refocused. When you have a smaller refocusing pulse, only a fraction of it is refocused. And these different signal pathways have different bold contrast. So these defacing pathways generate gradient echo like bold, which has a profile like vessel specific profile like we saw before. And we also have this refocusing pulse, refocusing pathways, which have a more spin echo like signal profile. But since we are acquiring everything simultaneously, the SSFP is usually the mixture of both gradient echo and spin echo. So like the contrast also depends on what is your TR, flip angle, and acquisition parameters and relaxation times. This determine how much energy is in different signal pathways. To make things more complicated or interesting, some of the bold is also stored in a longitudinal magnetization, which is decaying with respect to T1. And so this is like a quite basic picture, which gives you a basic intuition, but doesn't help you in real life. So most of the real life predictions are made with Monte Carlo simulation. So one of my colleagues now put up a open source Monte Carlo simulation, faster with the GPU and parallel implementation. So if you want to play with the SSFP or spin echo bold, you can look into this package. And so we want to do the SSFP at 9.4 Tesla, which is particularly hard, mainly because of the inhomogeneous B0. And but we also have quite a lot of positive things at 9 Tesla, especially the SNR and susceptibility. Since spin echo is like very much dependent on the susceptibility effect, it helps the SSFP bold as well. And and since we use a smaller flip angle pulses, we are like very much suited for for ultra high field than spin echo bold, which is usually very slow. And the typical bSSFP sequence looks like that. So you have a very fast repeating RF pulses and one short acquisition in the middle. So this most of the SSFP benefits come from this short acquisition, short repeating pulses. So unlike gradient echo, we have like much less T2 star blurring along the echo train, and then you have less distortion because there is no room for B0 inhomogeneity to play. And we also have a very high signal stability. So but the Cartesian SSFP sequence is quite slow, which is like 3 to 4 times lower than a gradient echo EPI. So we usually want to make this thing faster. So I mean, as said before, the contrast is also smaller, like we have only half of the gradient echo bold signal change, and we have bandings due to the B0 inhomogeneity and the acquisition speed. So we simulated the vessel specificity profile of different repetition time. As you can see, for 5 to 10, like the yellow line, have still some macrovascular suppression and a reasonable response for microvascular. So if you can increase the TR and then insert efficient readouts like a EPI or a spiral a trajectory inside, you get a very close acquisition speed as a gradient echo. So that's the goal of this project, and that's what we are trying. And when you have a non-Cartesian readout, you have a lot of problems, especially at high field, because of system imperfections like gradient and B0 off resonance. And therefore, we use an expanded signal model to reconstruct our image coming out of the scanner. And so we measure this gradient impulse response function with our scope camera, field camera, and we calibrate our coil sensitivity and field map, and we feed everything into an iterative inverse problem like the CG sense. And we speed that up with NUFFT and time segmentation for B0 corrections. And we can also reduce the number of iterations required with density preconditioning. So I don't want to dig too much into detail of the reconstruction, but just want to say it's available publicly. So if you have some issues, you can just see my implementation, which is like a combination of various open source project. So right now it's more or less stably, the image quality is more or less stable, but the reconstruction time is still 5 to 1 hour for each volume. So we usually do that in a cluster and do different reconstructions parallelly, which we can speed up with some fancy GPU reconstructions. So quick word about gradient correction. This is the acquired gradient impulse response function. In addition to that, we also acquire some filter delay. Like whenever you have a changing dwell time, there is a different data delay depending on the dwell time you pick. So we can calibrate that as well. So when your dwell time changes, the data is properly synchronized with your trajectory. So when you apply the gradient impulse response function, you pretty much see no change. So there is only like 1% of deviation. So that's shown in this orange line. And after gear correction, you have less than 1% of the maximum gradient. So this is how it is. So
if I may ask, this is a body gradient, right? Or
yeah, this is SE72, yeah, body gradient.
Gotcha, thanks.
And so since our trajectory starts from the center of k-space and it's very short, like maximum 6 milliseconds, you don't see a day and night difference between gear correction and nominal trajectory. So the most of the corrections comes from gradient delay, which is more or less corrected in nominal. So you don't see so much difference in a gear corrected image. So here you see a slight improvement. And for B0, like we have a acquired field map, which is smoothed, and as our readout is quite short, you don't see too much improvement in the passband where we are interested in. This only helps in the like the higher order passbands, I call them, where the B0 is much larger.
What is the MTI? That's like with the measured.
It's a time segmentation, yeah. So you have, I use different segmentation algorithm to speed up the B0 map because we are doing a 3D reconstruction. So you can't have a full signal model as it is. So you have to do a lot of tricks and to reduce the memory footprint for your reconstruction operator. So we use MTI, so you can use different segmentation algorithm rather than just expressing every single field map as a column of your encoding matrix. You can just reconstruct the images at different time segments and somehow calculate interpolation weights which sum them up together.
Okay, makes sense. So you never really used, like you said, you looked or you showed scope trajectories, but you're not really using them in your recon, right? Only to calibrate the GIRF.
Yeah, exactly. So the problem with that is scope has a also has an acquisition duty cycle limit. So like we are repeating at much faster pace, right? So we have a 10 millisecond interval excitation. So you can't measure the entire trajectory. Of course, you can measure for some time and fill the rest with the gear correction. So you have to somehow do some kind of blind spot interpolation to make this work. And you need a coil which can do that. So this we didn't had at the time. Yeah.
Okay.
Yeah. So it's just gear and B0 correction. This is very standard B0 correction. And so we did quite a lot of contrast like what is the relationship between echo time and repetition time on the bSSFP contrast, which I'm not showing here. I'm just showing the high resolution results. So all the experiments are performed at 90, and this is the sequence. It has 3 parts mainly. So pre-scan, and rather than removing dummies after reconstruction, we have a flip angle ramp to reach the steady state much faster, and you don't have to discard too many dummy volumes in the beginning. And this is the main acquisition. And these are the 3 protocols we tested. So here we mainly optimized for speed, and we didn't care too much about the SSFP vessel specific contrast or anything. So we applied standard full field visual checkerboard pattern with 20 seconds rest and 10 seconds stimulus. And then I think all of them are roughly 6 minutes of acquisition time. So all protocols just run 6 minutes, and it's a single run. So so just to see what is the functional sensitivity and stability of the acquisition. And as you can see here, we have a slightly different pulse, which is a binomial pulse. We use that for fat suppression because we have to always keep this pulse rhythm on. We can't have a saturation pulse like you do it in EPR. So we have to live with a binomial pulse. So the binomial pulse more or less works like that in ideal case, where you apply a pulse, wait for the fat to dephase pi phase evolution, and then apply the next half pulse, and then you get you bring the fat back to your longitudinal magnetization, and you excite your water alone. So so this very much depends on the off resonance of the fat. At 90, we have like 1.5 kilohertz fat off resonance. Therefore, like the interpulse duration has to be 0.36 milliseconds, which is not enough for gradient switching. So we went for a 3 pi phase evolution, which gives 1 milliseconds for us to switch our gradients for slab selection. And so you have a different frequency profile for different types of fat water excitation. And for a thin slab, we use a long water excitation. And we have a relatively long readout, so 6 milliseconds. So in 6 milliseconds, fat dephase quite a lot. Therefore, like you see a significant image quality improvement. And with the model reconstruction and water excitation, we can directly go into the high resolution protocol results. So we did the...
Maybe I have another question about your acquisition. So you're doing this at 9.4 Tesla, and are you using PTX or are you using CP mode? Like I know you have small flip angles, but still
Yeah.
it, like especially also the different paths that you mentioned might like push towards one direction or the other one based on your nominal excitation flip angle. What did you do for excitation?
For excitation, it's just a CP mode.
Okay.
So like we don't have any flash sequence which has, I mean, have a universal pulse, I think so. So far, I think we have only PTX implemented in MP ranges.
Okay.
But so this is like completely different developmental project. So we've been,
Gotcha. So not even kind of phase and magnitude shimming or anything like that, just CP.
Yeah, it's just CP, but I think it was quite optimal. I think you can't do much, or like, yeah, we didn't do B1 shimming for sure. Yeah. So the most of the time, the problem is you have to shim your imaging volume. So most of the time takes in shimming, try to see your bands, like where the bandings are, and then try to shim again, adjust your volume. So this is more or less first 20 minutes of acquisition is just going back from testing the sequence. We can't have a accelerated online reconstruction, so it's a basic gadgetron reconstruction. So reconstruct, see the bandings, adjust volume, do that again.
Cool, yeah, makes sense. Yeah, to focus on V0 more. Yeah.
So pressing issues first. So the so the first high resolution is 0.8 millimeter isotropic. We have a 22 millimeter thick slab, and then it's a 6-minute single run acquisition. And the main thing you can see is like there are no distortion. We don't do any affine transformation of our functional volume to match the structural. So it's just a reconstruction, and then we just overlay this MPRH image on top of it. And most of the activations are localized in the gray matter ribbon. You don't at this moment see any striped pattern or anything like that. I tried. There is first of all, it's a single run. There is a lot of low signal change. So there is roughly 3% signal change. which is half of the gradient echo, so you can't do much. So we at least try to smooth it a bit. So all the results I show have a smoothing which is equal to the resolution. So the full width of maximum is equal to the resolution to have at least some consistent activation across the gyri. And so the volume TR is 2 seconds, and we acquire for 6 minutes. And we did that for like 5 to 6 subjects. And you can see, like, if you have a favorable acquisition condition, like there is no banding in your region of interest, you see a very reproducible bold effect. And the same for 0.6 millimeter. Even at this resolution, we couldn't see too much of the striping you are interested to see, but we can image at this high resolution with a relatively short volume TR, and getting a reasonable activations was already quite a pain. And so some of the summary statistics, like of these two acquisitions, so to get an idea of how much functional sensitivity you're getting. So like the TSNR is actually quite low, which is very similar to gradient echo, where we expect a little more because of the SSFP. And for 0.6 millimeter, it's even lower. And this is the standard deviation and the distribution of your off resonance within your imaging volume. So for all this measurement, we use a TR of 10 milliseconds. So we have a passband of roughly 70 hertz. So if there is anything above 70 hertz, it will result in bandings. And this is the signal change distribution, and this is what most people report, the median signal change, which is roughly 3.4%. And for higher resolution, it's a little more higher. And at 9.4 Tesla, when you use gradient echo EPI, you have a signal change of over 8%. So it's only half of, little less than half of what you get. And the C-scores and the number of activated voxels are shown here because of the FSL statistics. It has this peculiar distribution. And we also tried a whole brain protocol, but with a lower repetition time for 5 milliseconds. So the acquisition is slightly slower than the 10 millisecond readout. But we can accelerate along both in plane and through slice. So we have a parallel imaging factor of 8, which reduces the volume TR to 2.88 seconds. So it's the same setup otherwise, 6 minutes, 10 seconds rest, 10 seconds stimulus. And because we reduce the TR, we also reduce the signal change, which drops to 2% from 3.5% a median. But otherwise, the activations are more or less localized in the gray matter. And as you can see, the bandings are quite visible even at such a lower repetition time. So we have roughly 150 hertz passband region. So at 9.4 Tesla, it's impossible to shim 150 hertz the whole brain. So you have frontal and temporal lobe bandings. So this is pretty much it. And the conclusion is, like, you have, when you combine these both, you definitely gain speed. So I did some literature survey, which is in my supporting information, which is based on Casper's 2D spiral, which claims to be the fastest spiral for some time. Then at least I at least want to beat him in one metric. So, and I compared across different SSFP and different encoding schemes. And then at least for the acquisition efficiency, we have see a clear improvement. And the other things like we don't have any distortions and the signal change, since we are increasing the repetition time compared to a conventional SSFP to 10 milliseconds, you get slightly better sensitivity. And of course, you lose quite a lot of things. The main problem is the reconstruction, which takes 1 day or in a cluster. And the banding. So if you increase TR, there is it's more frequency of resonance sensitive. So you lose the coverage at the cost of speed, and the TSNR is quite low, which which is not very clear because we expect a little more because of SSFP, and the iterative reconstruction also has some influence on the TSNR, which is not very clear. So, and there is also a little more T2 star weighting compared to a conventional SSFP because of the longer readout. This we have to take into account. And so the outlook, so we mainly focused only on the speed and a little less on SSFP and not at all on layer-fMRI. So we, in the future work, we would like to see some, to see the vessel specificity in vivo. So far we have only proven the vessel specificity in simulation and microsphere phantoms, but in vivo it's quite challenging. You need clever stimulus or different acquisition methods. This is what I'm trying now. So I will present something in ESMRMB in this matter. And yeah, so this is it. And just not to disappoint Renzo too much, I at least fished some images which is little related to layer-fMRI in that thing. So one of our, so from the 0.6 millimeter protocol, I calculated the signal change. As you can see, it's very noisy because of my thresholding. And we try to get layers from this MP-range images. So I tried both equivolume and equidistant layering after free surface segmentation. And as you can see from CSF to white matter, you see a similar drop, but the signal change is much lower. So for gradient echo, you have from 8 to 1% or something. Here you have much, at least the slope is much lower. And the orange line is the number of, it's a voxel histogram basically across the layers, just to have an intuition how much data I'm averaging across. And the same for equidistant, and you can see the voxel distribution is quite different, and therefore you see a little bump. I don't know whether it's a real effect or is it just a histogram. And few more things. So just to give you an idea about how gradient echo EPI looks at 9 Tesla. This is at 0.8 millimeter segmented EPI from 2D and optimized by our colleague. And as you can see, you get a very, it's a single run, 5 minutes, I think. And then you get a very nice, robust activation. And as you can see, the signal change histogram, you get roughly the median around 7-8%. So the same with gradient SSFP, you have a much lesser activation and the signal change drops to 3.3 or 4%. And but this either means SSFP is more sensitive, more specific, or less sensitive. So for this is the point we are in. We need further investigation to show whether this reduced effect is actually the macrovascular suppression. Would be nice to show. And one last slide, like where when we are in the piloting phase, we acquired a lot of images which we didn't present. And at that time, I was using a little longer stimulus, a 20-second rest and 20-second stimulation, same visual field checkerboard at 0.6 isotropic. And if you look closely, at least some of the activations have this not striping across layers, but has a hypointense region in the CSF region. So this is at least something to look forward to in the future. But yeah, this is pretty much it. And I would like to thank my co-authors from my group and the collaborators from DS Bonn, Philip and Martin. Thank you.
Thanks a lot, Praveen. That's really cool. I really like the quality of your spirals. Like spirals are hard and they're not very blurry, which I guess means that your B0 seems to work quite well. Or maybe it's also a matter of kind of your trajectory duration. So it seems that for each kind of shot, it only takes about 4 milliseconds or so, or 3 to 4, if I saw that correctly.
6 milliseconds now.
6, okay. So there's not a lot of T2 star decay during that. But this means you're doing kind of a multi-shot per KC plane. That's right.
Yeah. So it's segmentation along in plane, lot of spiral interleaves, and then you stack them to get a 3D K-space.
So what's the number? Like how many shots do you have per K-space plane? And I guess you do in the undersampling, you spread them out anyway.
Yeah. Like it's like at least it's uniformly spread. So if you, I think it's close to 15. I think
15.
For 12 to 15. I think like depending on the resolution. I think in the paper it will be written in my acquisition table, like how I partition different things. But yeah, so it varies depending on your FOV and the resolution and matrix size basically.
If nobody else has a question, I would just continue.
Actually, I was going to have a quick question, if that's possible. So maybe I missed it, but when you do the spiral readouts, do you keep your volume kind of aligned along the canonical XYZ axes, or do you tilt the volume?
It's at least I try to align along the Scalcarine sulcus. If whatever I can see from a localizer, I try to align along that. So it's for whole brain acquisition, it's transversal orientation. For thin slices, I try to avoid the temporal ear canals as well.
Okay.
So this, yeah.
Okay, thanks.
Maybe as you mentioned, the ear canals, they have B0 problems, but also those B0 problems are very, very locally confined. And with spirals, it's so tricky to get the right B0 reference map in your signal model, right? So you mentioned that you smoothed it, I guess, which I think is common as a regularization step, but you don't want to smooth it too much because then the very, very locally confined B0 problems of the ear canals are not accounted for. So how do you optimize your B0, which I found I heard is kind of one of the biggest limits in kind of the practical aspects of spiral reconstruction.
Exactly. So this had quite some problems. So the first I initially worked with phantoms, where I do this temporal. We have, of course, a lot of phase unwraps because of our 1 millimeter resolution. I think like you need a longer echo time, and then how do you correct for this unwrap is already a problem. So if you have a lot of fat in case of a human, then this temporal unwrapping doesn't work. Like at least first you need a fat fraction probably to separate things out before applying temporal unwrapping. So most of the, all of the bSSFP I acquire are the spatial unwrap, which already smooth things quite a bit. And then I use this regularized feed map estimator of like it's from Fressler. So I try to feed them in, try to optimize data at least once for the in vivo protocol I use, and try to use them. So it's not a super plain Gaussian filter, but it's locally, hopefully locally optimized to some extent.
That makes sense. Okay, cool.
So the other problem is if you include ear canal in your imaging volume, so the adjust volume should avoid it, and then you get more bandings in so the shimming algorithm needs little more smoothing to get this 70 Hertz frequency constraint. So this is quite hard to reach if you have a highly off resonant source in the adjust volume. So you have to be careful not to do that.
Does anybody else have a question? Otherwise, I would have a few more. Maybe you mentioned a kind of a setup with B0 shimming, that is like where you need to look at the data and the banding. You also mentioned that it takes about 5 hours to reconstruct one volume.
That's it depends on matrix size. So for smaller things like this depends on whether you need B0 correction or not. B0 correction is the most harder because you need to reconstruct at different levels, combine them, iterate, and then this explodes the reconstruction time. So what I do in online is just a simple gridding plus sum of squares operation. So I can't have acceleration while testing, otherwise you get bandings. And so this is like a very basic gadgetron implementation. So the main problem was aligning the data, like how to calculate trajectories and stuff. So we have a different package for calculating trajectories. So you have to embed all those things in your ISMRMD format. So this is the most hardest part. But then the spiral gridding and the coil combination is boilerplate gadgetron code. So, this is also shared along with the repos. If somebody is really interested, they can look into it.
Thanks. I really admire kind of your openness of sharing everything. Also, all the data, right? They are on Zenodo, and then all the hard part and the kind of the recon part is available on GitHub. Maybe I'm, which is a tangential question, but I'm maintaining, or I'm collecting more and more data on all the big labs doing layer-fMRI. Tübingen is one of the top 10 and in terms of publications, layer publications for labs in layer-dependent fMRI. But I could not find any kind of Cartesian EPI data from Tübingen. So you showed these Cartesian EPIs of the 6-minute experiment. They are not part of Zenodo. Are they? On your Zenodo, there's only the spirals, the non-Cartesian ones. Is there?
This was during the piloting phase. I just uploaded things which are for like right now we only have the time series of all the things and skull stripped T1 weighted image.
Okay.
And the raw data is already way too much to share. So if somebody is really motivated, we can arrange something else.
But there's no Cartesian sample of layer-fMRI. No. Okay. Okay.
I will talk to my colleague.
Don't worry. Don't worry.
If usually people are okay to share, because in Germany, like the data sharing is quite a hard thing to do. Right now, yeah, we have this. I think the slab things should be okay to share. I think so. unfortunately have to ask, and then usually people will share, I think, if they get some requests.
Maybe one comment about the layer profiles, which I liked a lot. You mentioned the kind of the binning of the voxels, how many voxels you have per kind of layer. You know, in Laney, there's this option, the flag of minus equal spins. Then you can basically flatten it out, and you determine the kind of the layers and force them to be the equal number of voxels per layer, if you want that. And
okay, yeah. Like as I said, I just don't know anything about layer-fMRI. Just followed some blog posts and just tried.
Yeah, that's really cool. Yeah, I like those profiles. So very often we are limited in EPI-based imaging with kind of B0 inhomogeneities due to distortions. Like, for example, if I'm interested in hippocampus or something like that, and which also has issues of large training veins. And so your method is kind of easier with respect to B0, but also harder, right? You have the bands, but at the other hand, you don't have all the EPI, like EPI artifacts that we do with respect to distortions, but also kind of the odd even delays and all those issues. So what would you predict? How hard would it be to do these kind of experiments further down the brain beyond V1?
The main problem is B0 problems. So the one other issue is like if you have a very short repetition time, the banding regions are more or less stable because we are like a breathing, like there's a lot of field disturbances because of breathing. So when you go down a lot, and then if you have a single band in your image, and if it moves too much, then you have much less coverage. But otherwise, if you, theoretically speaking, if you have very good field mapping down there, and you can remove all the bandings from your region of interest, I think it's possible. Yeah, field mapping and things gets harder, I think, down there.
Bella, do you have a question?
Yeah, this may have been covered and I just didn't hear it, but I was wondering, so your reconstructions, you're doing multiple shots of radial and then you'll kind of move through different segments. And those are all being combined as like a moving window, or those are done kind of as a block and then you move on to the next block? I didn't quite catch that part.
So we don't use anything like a view sharing, so it's just a single block. So we acquire a single volume, move on to the next whole volume, like undersample next volume. So there is no view sharing.
Okay, that makes sense. And I guess it's even more kind of a requirement to do that because of how computationally expensive the reconstruction is.
Like the reconstruction should be fine, I think. Like if you have a same sampling pattern, then it doesn't matter what data you put in. So if you arranging different volumes from view sharing is not super hard, but then you have intershot phase variations. This you have to correct for, maybe this will blow up your reconstruction pipeline.
Maybe I have a last question for me. Klaus Scheffler had these simulation papers looking at the kind of the non-steady state aspects of balanced SSFP. Also in Grace, usually people use these variable flip angles. It doesn't seem that you are limited by any of that, right? You're using kind of you had these dummies at the beginning, and you assume that they are basically taking care of all these transitions, and you don't have any delays, so you're just continuously scanning TR to TR. So you're constantly in a steady state. Is that right? Okay. Yeah. Okay.
So this is one of the reason why we are struggling with 3D. Like when you go for 2D, things gets, at least the reconstruction gets much easier, and a lot of signal instabilities gets easier. But since we have to maintain steady state across the entire imaging volume simultaneously, we have to go for 3D.
Good, makes sense. If there are no further questions, I want to thank you again. Thanks for this interesting presentation. And in 2 weeks, the next presentations will be by Daniel Hanold from MGH about EPTI VASO and machine learning vessel removal, I think, in fMRI. Right. Thank you very much. Bye-bye.
Thank you. Bye.